Great. So it's my pleasure to welcome you all to the Fujifilm Kickoff event. Fujifilm is our newest partner at MBC BioLabs. I know we have a number of internal attendees as well as external. If you're not familiar with MBC BioLabs, we operate premier life science incubator space within the Bay Area. Remarkably, we have 100 companies with us on our platform today. More than 200 companies have called MBC BioLabs their home. Those companies have brought more than 54 programs into the clinic and raised more than $5 billion. It's really a remarkable engine for life science innovation. That innovation is made possible through partners like Fujifilm. I'm sure all of us on this webinar know the name Fujifilm. Some of you may not know the extent to which Fujifilm has an existing and growing life science business, and that's what we hope to present to you today. It's my privilege and honor to introduce Aki Machida-san. He's a U.S. Open Innovation Hub Manager for Fujifilm. He'll tell you a bit about Fujifilm and then introduce his colleagues. After that, we have about 15 minutes for a Q&A. So if you have any questions, please hold them to the end. There is a Q&A button at the bottom of your screen. Put your questions into the Q&A chat there, and we'll get to them. So without any further delay, Aki-san, the floor is yours.
Okay, thank you, Robert, for the introduction of Fujifilm and myself. Today, I'm at the San Carlos 930 Britain building, and I'll take care of the opening and closing. As you can see, I have many presenters from Fujifilm's bioscience experts today. Sato-san and Alessandro from Life Science Cambridge office, Watanabe-san from Fujifilm Wako Chemical, Kevin from Fujifilm Irvine Scientific, and Liza from Fujifilm Diosynth Biotechnologies. Let me start off the corporate introduction. Next slide, please. This is a $20 billion corporate profile. It is publicly traded as the ticker symbol of FujiY. There will be a shareholder meeting on June 30, and the new chairman and the new CEO already announced. That will be officially effective. So you will see the big announcement right after the shareholder meeting at the end of this month. Next slide, please. This pie chart is the current business portfolio with the revenue and the proportional size. In the imaging solution, the legacy analog roll of photo film business is approximately 1% now. So over the last 2 decades, since the digital transition started, the original Fuji Photo Film Corporation has diversified into many areas and transformed their corporate characteristics drastically. And their corporate name has been changed to Fujifilm. We have done a lot of capital investments and collaborative business developments, particularly a heavy focus on our healthcare segment. The pie size of the healthcare business is currently at 26%, and we are expecting a higher growth rate in this segment. Before I hand over the talk to the healthcare representative, Sato-san, I want to conduct the polling question to the webinar participants. Billy, can you bring up the polling question? It doesn't take long, but it's a tricky question. Please choose the products and services that the Fujifilm currently does not provide. IPSL, microscopic camera, lab reagents and a test kit, contract manufacturing for antibodies, cell culture media. Okay. I don't know how long we should wait, but is it enough? Yeah, it looks like enough. So
Okay.
yeah, let's close there. So Sato-san will review the polling result. Sato-san, go ahead, please.
Okay. Thank you very much for your introduction, Aki. My name is Hiroshi Sato from Fujifilm Life Sciences Strategic Business Office. I'm here today to talk about Fujifilm's healthcare business, and particularly the segments that we call life sciences. Next slide, please. Okay, so as we just saw, about 1/4 of our current revenue is coming from healthcare related businesses. And when we look into this business segment, there are 3 pillars. The first one is diagnostic and therapeutic devices, including medical imaging, IVD devices, endoscopic solutions, which are used mostly in clinical settings. The second pillar, however, is therapeutic development, which is about drug development. Fujifilm is actively engaged in investigational drug development. We have drug pipelines in clinical stages in the US, Japan, and Europe for different indications that you can see here. And what it means here is that we are familiar with this process of drug development. We know what it is and what is required in different phases, which is connected to the third pillar, life sciences. And when we say life sciences, it is not necessarily about developing new therapies ourselves. It is about helping those who develop new therapies through our products and services. And that's what I would like to talk about today. And I would like to pause here and revisit the polling question that we saw earlier. Billy, could you bring up the polling results if you could? Okay, thank you very much. So again, the question was, which of these products and services Fujifilm does not provide? And we provided 5 options. All these results are very interesting. The correct answer was number 2, microscope and microscopic cameras. Congratulations to those who got it right. I'm not giving you anything, sorry, but it was number 2. So we recently took a brand survey and found out that Fujifilm as a brand is very closely associated with microscopic products, even though we don't have any of them. I think it's in analogy with companies like Nikon, Olympus, like camera companies having microscopic products. So it may not be surprising for you, but it was surprising for us, and recent findings again. Could it be a potential expansion area for us? Maybe, maybe not. But what we do not have is not so important here. What's more important is what we do have. And the fact is that Fujifilm has all the other 4 options that you saw, as well as other offerings as part of this third pillar, life sciences. Next slide, please. Okay, so our mission in life sciences is again about supporting, accelerating transformative life science breakthroughs. We want to help bring new therapies to the market, to the patients. We want to make it happen and make it happen earlier and in a more efficient way. And although our products and services are, I would say, primary means of achieving that goal, it does not end there. And I would like to present one example in the next slide. Next slide, please. Okay, so we started this Fujifilm Fellowship program at Harvard Medical School, which has supported 11 scientists so far. And we started this because we believe that it is important to start supporting the therapeutic innovation from very early stages, which is basic or translational researches. We believe that this research could make huge impact on therapeutic technology, therapeutic landscape. It may take 5 years, 10 years, 15 years maybe, but we want to make that difference because we are committed to our goal. Next slide, please. The partnership with MBC BioLabs is also one of the steps in that initiative. This community of of companies with the potential to entirely change the therapeutic landscape. It is supporting such a community as a sponsor, as well as working closely with companies like you, is very closely aligned with our mission, again, in the life science space. And that's why we are here. Next slide, please. Okay, so where to talk Fujifilm, where to talk with Fujifilm? I would like to suggest two options. One option is Open Innovation Hub, which is a division of Fujifilm led by Aki Machide, whom you saw earlier. It is located in the West Coast and is in charge of broader industrial outreach. And the other option is Life Science Strategic Business Office, which is my office. And it is located in Cambridge, Massachusetts, and is specialized in life science businesses. Next slide, please. Okay, contacts for general inquiries. Again, I suggest reaching out to Aki, who is at Open Innovation Hub USA. But for more technical or scientific inquiries, if you have any, I would like to introduce one of my colleagues, Dr. Alessandro Amici, who is our Senior Business Development Manager and has cell biology background. He is joining this webinar as one of the speakers as well. So if you have any questions, please feel free to type them in the Q&A box so that we can address them. Next slide, please. Okay, that was the end of a quick introduction corporate presentation part. And before moving on to the next section where we make a deeper down into Fujifilm's life science offerings, I would like to give you a quick overview of, again, who we are and what we do in this space. Our goal, our mission here is to support therapeutic development through all stages and steps. And as you can see, it spans from very early phase of drug discovery through preclinical development and clinical development to approval launch and commercial supply. You can see different lines of products and services and different brands under Fujifilm umbrella with the same mission in mind. I think maybe some of you recognize these brands. I hear that some of the companies at MBC BioLabs worked with Fujifilm Cellular Dynamics, which is a leading supplier of human iPS cell products. But due to time constraints, we are going to introduce just 3 brands out of these. The first one would be Fujifilm Wacko Group, which is specialized in laboratory reagents, laboratory automation systems, or quality control testing solutions. The second, Fujifilm Irvine Scientific, a leading provider of psychomedia media development and optimization services. And the third, Fujifilm Diosynth Biotechnologies, a contract service provider for biologics, virovectors, and vaccines. So to begin with, I would like to hand this over to Dr. Mitsuo Watanabe from Fujifilm Wacko Chemicals USA. Mitsuo, are you ready?
Yep.
Okay, here you go.
Yeah, next slide, please. Hi, my name is Mitsuo Watanabe from Fujifilm Wacko Chemicals USA. I'm going to talk about our company, one of the life science subsidiaries of Fujifilm. Next slide, please. Our company originated from pharmaceutical merchant business, started in Osaka during the samurai period about 240 years ago, which is now Takeda Pharmaceutical. This picture shows the old city of Osaka in the samurai period, and you may be able to find Osaka Castle somewhere in this picture. Next slide, please. In 1922, the chemical department of Takeda Pharmaceutical was separated to start the reagent business to serve high-quality chemical reagents to the researchers in Japan. After World War II, the company name was changed to Wacko Pure Chemicals, and we have been supporting academies and industries to by providing high-quality reagents and services inside and outside of Japan. In 2017, we joined Fujifilm Group and our company name was changed to Fujifilm Wacko Chemicals Corporation in 2018. Next, please. This slide shows our 3 core business areas: laboratory chemicals, specialty chemicals, and clinical diagnostics reagents. Next, please. So we manufacture a variety of the products at these manufacturing sites located in Japan. And we have R&D facilities near Osaka and Tokyo to develop new products and technologies in collaboration with researchers around the world. And headquarters is located in Osaka. Next, please. Oh, yeah, and the manufactured product in Japan are distributed to our customers through our strong distributorship network, including us, Fujifilm Wacko Chemical USA. So this picture shows our company's campus located in Richmond, Virginia. This area includes FDA-registered IVD or biologics manufacturing and distribution site. Next, please. So this slide indicates that our research use reagents to support life science research, as shown. So as you can see, there are many categories of the reagents available, like antibodies and ELISA kit of your interest. And you can find the unique reagents from our website. Next slide, please. So I would like to show some of our focusing research area of our products. So as you may know, there are significantly increasing activities in exome research. Exomes are the nanoscale particles secreted from almost all cells tested, and they pass the variety of signaling molecules from donor cells to the recipient cells. Researchers in this field are studying the biology of the exomes and the therapeutics and diagnostic applications of the nanoparticles. Next slide, please. We have a unique product to isolate exomes in high purity. Conventional purification method is ultracentrifugation, but it precipitates many contaminants together with exomes during the purification steps. Our method utilizes a calcium ion-dependent binding between TIM4 protein and the phosphatidylserine on the surface of the exomes. The captured exomes can be released by a mild condition just by adding the chelating agents such as EDTA. So highly purified intact exomes can be isolated by using our kit. And you may be able to use such isolated exome for more precise functional and omic analysis. Next slide, please. We also have the reagent for the quality control. And this is example for the detection of toxin, which is a polysaccharide, is a component of the gram-negative bacteria cell wall. The medical devices and pharmaceutical manufacturers need to control endotoxin levels in their products. We have a lab reagent which are prepared from the lysate of blood cells from horseshoe crab. And the enzyme cascade shown in this slide is utilized to detect a tiny amount of endotoxin. Next slide, please. So here are the reagent and the instrument for measurement of the endotoxin, and we provide these devices to the global market. Next, please. We also supply some cell culture media and additives for cellular biology research. So these pictures show that our neuron culture media supports maturation of the neuron cells, better than the competitor's product. And we have many product line to in this field as well. So next slide, please. And we have many IVD products. And this slide shows one of the example of our unique technologies. We have developed and launched the IVD medical devices for the liver cancer biomarker, FLC, by using microfluidic affinity electrophoresis. During the electrophoresis, the migration of the lectin-reactive AFP isoform, which is the liver cancer-specific protein, is retarded by interacting with LC lectin in a channel. And it can be separated from non-malignant AFP isoform, AFPL1. And we can use this AFPL3 as a marker for liver cancer. And this product are FDA cleared and utilized in the United States and other countries. Next slide, please. So this is another application of microfluidic technologies for the molecular IVD testing. This microfluidic chip in the middle of the slide has the PCR chamber and the separation channel. So PCR chamber is utilized to amplify the pathogens, target DNA or RNA, and amplicon is separated in a tiny microfluidic channel on a chip and quantify the amount of the amplicon, and which corresponds to their pathogens and pathogen amount. And you can get results from the sample in a short period of time by the automated instrument, Mutaswap or G1. For example, in the case of Mycobacterium tuberculosis, it takes only 45 minutes from the sample to answer. So next slide, please. We also have some automation solution and software solutions to support high throughput research needs in life sciences. Next slide. So you may find more unique research use reagents through our website, and we will be happy to supply reagents and chemicals of your needs. Okay, thank you very much for your attention, and I'd like to pass over to Kevin from Fujifilm Urban Scientific.
Great, thank you. Next slide, please. Good afternoon, everyone. My name is Kevin Colell. I'm Global Marketing from Fujifilm Irvine Scientific. I want to thank you for joining us this afternoon. Next slide. So just to give you a bit of an orientation around our company, Fujifilm Irvine Scientific is divided into two divisions. One is the life science division. The other one is our medical division, focused on assisted reproductive technologies and cytogenetics. For today's purposes, I'm going to focus on the bioproduction and cell and gene therapy segments. And as a level setting, bioproduction for us includes any of the clinical, commercial biologics or therapeutics, such as monoclonal antibodies, recombinant proteins, viral vaccines, and then for cell and gene therapy. Our primary focus and what we're known within the market is being an extremely flexible and responsive partner. Our primary goal is to assist you along the way, whether you're a small startup organization, you're looking for the speed element to get your new innovation to market as quickly as possible, or whether you're a multinational with a rich pipeline driving down the accessibility and the affordability of biologics. Our primary focus is to start our conversations with understanding your high-level strategic goals and then building our portfolio and our capabilities around you to enable you to realize your vision for your company. Next slide, please. So as a high level overview of our key products and services. We offer both off-the-shelf products as well as our services and custom manufacturing. So starting at the left, we offer cell culture media supporting CHO, HEK293, and insect cell lines. We include in this our Prime XV portfolio supporting stem cells and primary cell lines. I'll speak a bit more about those two aspects as we go along. But another part of our offer is our services offer. And so, for example, our Express Media service allows you to have your development formulation or your prototype formulation manufactured in 10 working days or less from receipt of PO so that you can get your innovation to market. We also have within our various sites around the globe, media development optimization scientists, and they are able to engage with you in consultative work or actually receive actual cell banks for doing media development and optimization per the performance requirements that you're looking for your therapeutic or biologic. In addition to that, we are constantly launching new prototypes for the various cell lines, and you're welcome to try any of our survey panel supporting CHO, HEK293, and the like. And the last part of our offer actually is the biggest part of our business, which is our customs. We offer large-scale cGMP manufacturing and various packaging, various formats such as liquids and powders suited to your needs on C of A or specifications that you may need going forward. Next slide, please. So a typical pathway of working with our customers follows this. Customer has a early stage clinical program that oftentimes they have their own media formulation. And with each new program and each new cell line, we know that cells have different nutritional requirements. And so, like I said, on our Express Media service, we're able to manufacture that your custom formulation with your tweaks to enable scalability of that media formulation. So once you identify and lock that formulation, you know that it's scalable to large size manufacturing. In addition to that, our media development program enables that optimization, whether you're looking for critical quality attributes or titer levels or longevity. Our media development scientists engage in you in various programs. Many of our customers not only explore their formulation, but depending on the program timeline, also compare it to what's available in the market. And so, as I mentioned before, our balanced CD platform is often compared to an existing formulation. And again, we offer the ability to adjust and modify that to suit your needs. Our whole goal here is to lock that process so that you can go ahead and file with the regulatory bodies and go ahead and commercialize your innovation. Next slide, please. So starting with our biologics production platform, this is our balanced CD cell line. So it's media and feeds, both within the fed batch and perfusion development processes. Some of our best performing media are chemically defined media. This enables an easier pathway towards regulatory approval. One of the key aspects that we focus really hard on is the raw material control. So media is typically made of 70 to 100 different components from multiple vendors. And so reducing the variability by controlling that raw material input is very important for us and for you. So we try to control that as much as possible. As I mentioned, we have cell-specific media supporting CHO, HEK293, and then Vero and MDCK cell lines in different packaging configurations, whether it's a small bottle for discovery reasons, or possibly it's a disposable bag for cell line development through manufacturing. Our powder formats allow us to package in large production capabilities up to 50 kilogram sizes. And we actually have one of the largest blenders in the industry to make 7-ton batches of media, thereby again reducing the variability in your process so that you can achieve that affordability and accessibility of your biologic. Next slide, please. Our cell and gene therapy platform, again, is a media supplements providing seamless workflow and solutions. We employ our same expertise internally to develop these end-to-end solutions with similar raw material control. What's a little bit different about this part of our portfolio is we're actually able to leverage some of the needs from our medical sides who have very strong regulatory interface with the various bodies around the globe. So this portfolio, we're working very closely with the regulatory bodies to make sure that as you spec in a media into a cell and gene therapy, which is closer to the patient, there is a good safety profile and it will meet the regulatory needs for the various government bodies. This also is scalable to commercial volumes, and we offer cell-specific media. So whether it's mesenchymal stromal cells, hematopoietic cells, T cells, natural killer cells, or dendritic or neural cell lines, we have media appropriate for each one of those. I should also say that we are customizing and working towards driving a closed system, closed automated system, as many of these cell and gene therapies use different cell expansion systems. A key requirement is to have that closed-ended automated platform. And so we want to make sure that the raw material, the buffers, and the processed liquids are inputted seamlessly into that workflow. Next slide. So I want to highlight a couple of our key flagship products. The first one is our T-cell chemically defined media. It's the only one on the market in terms of chemically defined, supporting rapid T-cell growth and viability while maintaining the functionality that you require. It supports T-cell expansion, both from PBMCs as well as isolated T-cells. It's chemically defined, containing a recombinant protein source, animal component free. It does require the various cytokines that you may need for your end user application. And I should also mention a key part of this is, again, that ability to help you define your process. So we have many publications, presentations, and protocols available for discussion, and actually a key field application scientist to work directly with you to define and lock that process that you want to take to scale. Next slide. A second key product for us is the PrimexV MSC expansion xenoserum-free media. This is xenoserum-free media supporting different tissue lineages, whether it be bone marrow, adipose, or umbilical cord tissue. It's a complete, ready-to-use media, so it simplifies your workflow. It can be used without attachment substrate, although we do advise optimal performance through various attachment factors. As I said, serum and xeno-free, animal component-free, and this is also supported through various publications and posters and protocols with our field-based team. Next slide, please. And the last slide I wanted to introduce is innovations are only as good as getting them to market. So one of the things that we've spent a lot of effort and continue to grow and invest our capital in is building out our global footprint. We have sites in California for liquid as well as powder manufacturing, a site in Netherlands that is in development due to open at the end of this year, supporting powders as well as liquids, and then a site in Japan as well. What's key to this is that we have seamless scale up or seamless supply across these sites. So we identify that the same raw materials, same equipment, and same SOPs with the same qualified, certified quality systems is important for your business continuity. What we like to show here is that we're able to support local supply with our global partnership, and we look forward to engaging with you. Next slide, please. And with that, I thank you for your time. I'll turn it over to Liza for Dyson. Thank you.
Hello, good afternoon, everybody. I'm just gonna wait for my slides to come up. My name is Liza Rivera, and I am responsible for marketing and communications at Fujifilm Biotechnologies. And what I'll do today is just give you a brief introduction. We are strictly focused on offering services and solutions via services to the biopharmaceutical industry. We'll talk a bit about those. If I could have the next slide, please. So it's interesting when we did the survey that 33% of the respondents did not think that contract development and manufacturing work was part of the Fujifilm portfolio. And we are part of that division. As a matter of fact, Fujifilm announced back in 2017 that they were launching what they call the Bio-CDMO division as a strategic business unit, and we are one of the big components of that division, and one of the big commitments Fujifilm has made as an organization is to really grow the contract development and manufacturing side, which you can see by the number of investments that has been announced, that we have announced in the past 24 months. But I think it's really important to mention that there's a really big level of commitment behind what we do. And we are really delighted to be offering our services and the solutions and working with various companies across the world to bring therapeutics, vaccines to the clinic and hopefully beyond. Ultimately, it's all about making things happening for patients. We offer this, we have a global footprint, so we have locations across the world. In the United States, we have a facility in North Carolina, we have a facility in Texas, and then in Europe, we have a location in the United Kingdom, in the northeast of England, and we recently acquired a site in Denmark, a former Biogen facility, which is now a Hilleret facility for large-scale cell culture production. We work in multiple modalities, so we can offer solutions from your traditional cell culture, microbial, and with the Texas facility in 2014, we entered the gene therapy space. So we have gene therapy, viral vectors capabilities as well. And one of the things is that we will, and I'll show that in a bit, I think is the next slide, but one of the things is we can support our customers from fairly early in the beginning as they're developing their therapeutic candidates all the way to commercial production. And we bring that experience across all of our sites. We have been licensed to manufacture 16 commercial products throughout the world. It's a mix between cell culture-based products and microbial products. And sometimes the numbers change a bit, but it's about over 30 years. So we have completed over 500 different discrete projects across. And right now, in any given year, across all the sites, we're working in a very large number of various programs. As of when I put the slides together, it's 138 active programs in one way or another. And you may be wondering, how is that possible? But if we go into the next slide, no, it's going to be the next one, but I'll come back to that. You'll see where in 138 programs, but we offer a broad range of services, and you can see why we have so many different areas. I'm not sure how many of the audience is familiar or has worked in the past with a CDMO, but I just wanted to touch briefly on that and what it's like to work with a service provider and with us. So we strictly focus on development and manufacturing services, and everything we do is for third parties. We don't develop anything for ourselves. We don't market or commercialize any products. Everything that we do, it's in that space, and that's what we have been for over 30 years. So that's what the foundation of our business. We work. We can start, from the very beginning, where you don't even have a cell line to express your molecule, and we can help support that. Or you have already developed a process and you want to optimize it for scale up, or you already have a process and you're running it somewhere else and you're looking for security of supply. We can do that too. So we work with customers that have really are very different and they can come to us at very different stages of the clinical path. Some customers, select working with a CDMO for different reasons. Capacity and technical expertise, capabilities is always really important. Capacity is being able to get in and get your product when you need it. Compliance with its track record, and that goes back to the many commercial products that we have. So we have a very robust and excellent track record when it comes to regulatory inspections, and not just the inspections from multiple agencies around the world, but also we are inspected almost every week at every site by potential customers. So we're very proud of our quality. And one of the things that we know when customers come to work with us is, how can we work together, and having a partnership like type of agreement rather than strictly fee for service. That's very rigid, and we want to say that we offer flexibility to be able to work with our customers and deliver what is needed when it's needed. What is really neat about being in a CDMO, and I have personally been in the company for about 13 years, is because we work with so many molecules, we see pretty much everything from pretty straight monoclonal antibody processes to more complex types of molecules, and all of that knowledge and expertise, it's what we offer to everyone, because we've seen if there's a problem, or there's a not like the word problem, but if there's a challenge when you're developing your process or optimizing your process or troubleshooting through something, it's likely that we have seen it. And we have over 500 PD scientists doing this work, and it's a great learning place, and it keeps us really current. So those are some of the reasons why working with a CDMO and how we work, and what some of the expertise that we gain from working with so many products, which is then translated to what we offer in our scientific excellence and expertise. Next slide. So this is the slide I was trying to get to. My colleagues have talked about where they fit within the development process or the clinical development journey. We offer solutions all throughout, from preclinical all the way to regulatory approval, launch, and then just supporting the life cycle of a commercially available product. I'm not going to read all of these services, but we start pretty much with expression studies. So in most cases, when we are at this stage of conversations, customers have an idea of how they want to express their molecule, but sometimes they don't. And they might be considering different options, and that's something we can do, or we have platform technology in cell culture called Apollo X. We also have a microbial expression platform called Paveway. So we can work really early. We can develop the process, from the cell line or the strain, and we can develop a process de novo, and we can at this stage also support production of some material for toxicology studies, etc. We do a lot of process development, process optimization. We have a lot of expertise in analytical development. We can run stability studies for both drug substance and drug product. And then, of course, we have cGMP manufacturing capabilities across the world. I'll show you a bit of where we have that capacity and at what scale. And we have now added as well fill finish services, fill finish offerings. And we also have the ability of doing assembly, labeling, and packaging, which is a new capability that we got from our Hillera facility. And we can do this. And then as you move through the different clinical stages, we can support it. And sometimes, customers come and work with us, and then they go do their clinical trials and they come back. Sometimes they sell the product and then the product comes back under a new owner. So it's just really interesting. really important is that as we're working with you, one of the key things that we want to offer, particularly in the development of a process, is we want to develop processes that are going to be pretty robust from early on. So it's going to be a process that is going to get you from phase 1 to commercial without having to do too many changes, or it's all about manufacturability and making sure that you can scale up a process to support the demands of your patient population. We have a lot of expertise in other aspects, which is the process characterization now that's moving into late phase and preparation for your PAI, process validation activities, and then of course we support all of the post-approval product lifecycle management activities once a process is commercial with us. So we can help you, we can offer a solution and partner with you pretty much anywhere you are in the clinical development process. Next slide. This is just a slide to highlight some. This doesn't represent all, but some of the regulatory agencies that have visited our facilities. Next slide. I'll just talk a little bit about, just give you a little bit more information about our different core businesses. We are one of the leading microbial fermentation CDMOs in the world. This has been part of our DNA, if you will, from the very beginning of us as an organization. And we've worked in these numbers are here, but we've produced way more than 2,000 GMP batches in our history. You can see some of the microbial processes that we have developed and then translated into GMP production. We currently have 4 commercially approved products that we have developed and then transitioned into manufacturing. And one of the things is, I don't have a slide about this, but it's very important to mention that as an organization, we have a huge push for innovation work. And we actually have a dedicated initiative internally. And the paveway expression technology, which is based on E. coli strain, and it has 3 parts. We have host lines that we have identified as high producers, and we do have some proprietary vectors that we apply. And then we have the process development part component of the whole platform. But this actually, paveway was something that came out of innovation initiatives. And what is really neat about paveway is you can do an evaluation of your molecule and see if it's going to express well using the paveway technology in 4 weeks. Fermentation processes are very fast, and we have really set this up to be able to tell you, yes, we can get really good expression using paveway, or maybe not quite as well. And we can also let you know how what type of expression is going to best work for your molecule with paveway. If we go to the next slide, we can talk a little bit about our cell culture experience. And we have the counterpart to paveway, which is Apollo X. This is our advanced mammalian expression system. This is a CHO-based cell line, DG44. And it is, again, it's a platform that we have, so we can start working with customers really early. We can get about the expression with Apollo X is between on average 7 to 10 grams per liter. And we have a pretty good sense of how Apollo X is going to perform 10 weeks after transfection. Again, Apollo X is part of our internal initiatives. It was developed in-house. We've done over 25 cell lines have been created. We have a lot of expertise in cell culture, so from monoclonal antibodies, standard CHO programs, as well as other recombinants. And what is really interesting is we have a lot of expertise in the baculovirus systems, and not only developing processes, but actually taking those processes into GMP and scaling them up for commercial production. And we have been heavily involved in the manufacture of some of the COVID vaccine candidates, and some of them, one of them, the products that we're working on is a baculovirus express vaccine. I'm going to move really quickly because I'm running out of time. If we just go to the next slide. We offer advanced therapies, viral vaccines, and gene therapy services. And this again happens in our Texas facility. What I really wanted to show here is that we have experience working in a wide range of expression systems, viral vectors, and we also have worked with a significant number of virus replication platforms, and we can bring all of these multimodal modalities into our facilities in Texas. And now we're expanding to Boston and also in the UK to offer these services. And then the next set of slides, if we can just go through those quickly, talks about our manufacturing facilities. Actually, this is a really nice summary slide, but we offer capacity across the world, going from 200 liters all the way to 20,000 liters in cell culture, going from 100 liters in microbial to 5,000 liters, having multiple units, and then having a wide array of capacity for the viral vector system. So if we go to the next set of slides, it's just going to show exactly what our capacity is in North Carolina, and we can make this available if anybody in the audience is interested in seeing this. And then the next set of slides, you can just go through them because it's what we offer at the different sites. And that's what we do. So with that, I appreciate your time and your attention. Thank you.
Thank you, Lisa, and thank you to all the presenters so much. We covered a lot of ground really quickly. Taking a look at my notes, we talked about exosomes, microfluidic systems, high-performance cell culture media, cell and gene therapy manufacturing, and end-to-end CDMO. So thank you so much for running through all of that so quickly for us. Of course, I'm sure attendees have questions, but not to fear. Aki Machida-san has an office here with us at MBC BioLabs. You can come find him anytime. He can connect you with the right people within the Fujifilm family to get your questions answered, or please just come to any of us at MBC BioLabs, any of our team, and we'll get you connected as well. So we have just a few minutes for questions. There is a Q&A button at the bottom of your screen. Feel free to put a question in there, but I'll kick things off just really quickly. Obviously Fujifilm has a long history in film manufacturing and developed a lot of expertise and technology around film manufacturing. How has that been applied sort of broadly to the life sciences? Any of the panelists, feel free to answer.
I'm happy to jump in there. If you think about film manufacturing, and I don't know how familiar people are, but you have to be in clean rooms. It's really controlled environment. So when you think about film production, it's really not that different from the production that we do in a GMP environment. So a lot of that operational excellence and working in these rooms and understanding the high standards of quality we have really are very compatible with what we do coming from Fujifilm Corporation. And we have incorporated many of the operational excellence learnings from the film industry into what we do. So it's just, there's actually more parallels than people are aware.
Right, that makes sense. And then, given the several different brands that Fujifilm has in the life sciences, how do those come together collectively to provide a value proposition?
I think I can address that question. As we have seen, we have different brands and product lines through all stages and steps of therapeutic development. And I think what we want to be in the future is a one-stop solution provider. And as Liza addressed in her presentation, we want to be a partner for biotech companies which can work with them through these processes from very early phase to the commercialization. And as I explained in my slide, we have an office based in Cambridge, Massachusetts, which is in charge of kind of connecting these different divisions and brands so that we can offer more one-stop experience for customers. And I think that's the direction we are heading in terms of total value proposition for customers.
Makes a lot of sense. We have a question from the audience, and it's related to cellular dynamics. We actually can't answer that on this presentation today, but I'll definitely get back to you and connect you with the right people at Cellular Dynamics to answer that question. And we're running short on time, so Aki-san, perhaps I could put it back to you for a few concluding slides.
Okay. Good. Well, thank you very much. So, Billy, can you pull up the last few slides? After this. Yes. So, well, thank you, Robert. It was a nice discussion. There are 3 more quick slides to close today's session, so please don't go away. Open Innovation Hub activity has started at that facility in Santa Clara, California. And you can actually take a look at the inside briefly by 3D virtual Matterport service. As you have seen in the corporate business pie chart, each table is dedicated to each business segment, such as inkjet printers, display materials, imaging products, data storage solutions, and Instax cameras. If you are interested in visiting this facility, please let me know. Next slide. Next slide, please. So to conclude today's event, we would like to thank MBC facilitators to make this happen. For those who want to evaluate today's session or connect with us later, please fill out the follow-up survey. Also, if anyone is interested in adding their picture on their ID card holder like this, we are happy to take a picture for you by Instax mini camera. I and my staff will be at the cafeteria this afternoon, and also normal business days, or someone should be in the room 311 of 930 Britton Building. Again, thank you very much for your time with us. Thank you very much to presenters, and thank you very much. See you again soon.